Hey, welcome back to Academy Idol. Today I want to talk about extracellular aggregates, or junk as we can call them. The junk between our cells are proteins, but we generally call them amyloid. Amyloid becomes a big problem once it starts sticking together and interrupts homeostasis. Beta-amyloid is actually the most popular type of extracellular garbage. Beta-amyloid being most devastating once it starts forming between neurons, impairing a wide range of brain function, memory loss being the most noticeable. So another example of proteins sticking together is called the islet amyloid. To elaborate a bit more, islet amyloid is actually associated with type 2 diabetes. However, it's not very clear whether or not type 2 diabetes is caused by the islet amyloid, or if the islet amyloid is a consequence of type 2 diabetes. Senile cardiac amyloidosis is another disease in which amyloid plays a significant role. In this case, it builds up in cardiac or heart tissue. In most cases of this disease, it's very fatal. There's actually some evidence that suggests that cardiac amyloidosis is one of the major causes of death in people over the age of 100, which is kind of interesting. So amyloids are a big problem. How can we start removing them? We can actually get our immune system to attack it. If we can get antibodies for the specific amyloid, then we can stimulate our immune system to build up defenses against it. There are two ways we can do this, either the passive or the active method. The active method is actually to stimulate the immune system to build antibodies for the amyloids by injecting tiny fragments of amyloid into the human body. So the passive way in this case is actually to create the antibodies outside the body and then just inject the antibodies directly inside. So this seems very good, but there are some problems with it. When the amyloid antibody complex tries to cross the blood-brain barrier, for instance, it might get stopped. So there is actually a more promising method to get rid of the amyloid between our cells. So this technique uses catalytic antibodies. Catalytic refers to catabolism, which means to destroy. This is actually a very efficient way to do the job because catalytic antibodies can destroy amyloid and just keep going over and over and over. Other types of amyloid have a limit. They get used up easily, therefore they require larger doses. Not only that, but catalytic antibodies come from a class of proteins that have an easier time passing the blood-brain barrier, so you got another benefit there. This is highly important for scientists trying to study Alzheimer's or try to cure it because they're normally targeting beta-amyloid, which exists inside the brain. Also, because they're catalytic antibodies, they're going to be obliterating the amyloid, breaking them up into very small pieces. This means you don't have to worry about trying to transport all of that amyloid, like the entire structure, past the blood-brain barrier. That would prove to be far more difficult than just breaking them up into very tiny pieces. So in summary, essentially to rejuvenate our body in this context, biologists are going to have to develop vaccines that can destroy the amyloid inside our heart, brain, and circulatory system. And the best way to do this is to use catalytic antibodies, which can destroy the amyloid, or any other technique you can come up with which would break apart the amyloid. This way the body doesn't have to worry about transporting very large amyloid molecules outside of the area through the circulatory system, which could cause more blockage. So that's basically it. I hope you learned something about extracellular junk or amyloid. Please like the video to show support and subscribe for more educational videos regarding human aging in most cases. And as always, thank you for watching.